In this video, I want to talk about the process of cell division known as mitosis. There actually are two types of cell divisions that are used to create more cells. Mitosis, the focus of this video, creates two identical daughter cells from one parent cell. And the daughter cells contain the same amount of DNA and are known as diploid cells. Mitosis is used both by prokaryotes and eukaryotes. Unicellular eukaryotes use it to reproduce the entire organism. And for multicellular eukaryotes, mitosis is used to take that organism from a single fertilized cell at the beginning of its growth and development to the complete organism, which will have many, many more cells. Mitosis is also used every day in organisms as they stay alive to repair any damaged tissue or to replace worn-out cells. The second type of cell division, known as meiosis, creates gametes or sex cells, such as sperm and eggs, in multicellular organisms that reproduce by sexual reproduction. So the daughter cells created by meiosis contain half the DNA as the parent cell, and in that case, they're called haploid cells instead of diploid cells. In sexual reproduction, of course, two of these sex cells, two gametes, will combine to create a single cell known as a zygote that has all of the same DNA as the parent, and then that one cell will use mitosis to grow the organism. We'll be talking about meiosis in a later video. So in mitosis, one cell becomes two. It begins within the nucleus of the parent cell and actually has 5 distinct stages that you'll need to be able to identify. Mitosis, as I said before, ends with the formation of two daughter cells that are identical to the parent cell. In fact, they are clones because they have the same DNA, so they will have all of the same genes. The first phase in mitosis is interphase, and really this is where cells spend most of their time. Interphase has when the cell is carrying out its ordinary activities. During interphase, the DNA is loosely coiled. We call it decondensed. And in that form, it's known as chromatin. It has its own specific name. And chromatin fills the nucleus with no distinct areas. When it's stained, it's basically stained all one color. You cannot see any chromosomes. There are no distinct chromosomes. DNA replication, which is necessary before mitosis can occur, will happen during interphase. And if you look, this diagram to the right is known as the cell cycle, and it's taking a cell through its lifespan where after a new cell is formed, it sort of starts here, and we see some cell growth happening during this time period, and then DNA is replicated to prepare for cell division. There's sort of a final bit of growth, and then mitosis happens in this short period of time compared to all the other hours that this cell is spent alive as the cell divides. So as I just said, when the cell is in interphase, the DNA is found in a form that we call chromatin, where it's very loosely coiled or decondensed, kind of like a plate of spaghetti. And it will duplicate itself, and the original strand, say this is our original strand here on the outside, and the duplicated strand are held together by a structure called a centromere. Then these long strands of chromatin will curl themselves up to make the coiled form or the condensed form that we call a chromosome. But this chromosome is actually made of two identical strips of DNA, which in this case are called sister chromatids. And then the whole thing together is a chromosome. So we have chromatid, chromatin, chromosomes are all referring to the DNA in different stages of mitosis. So once interphase is over and the cell is moving into mitosis, the first thing that happens is known as prophase. And this copy DNA coils up tightly so that we can actually see visible chromosomes. So this is just a diagram, but if you're looking at the cell under the microscope, you can see the chromosomes in the nucleus. Because these chromosomes are replicated and they're going to have to be divided into two daughter cells, the nuclear membrane, which is also called the nuclear envelope, has to break apart, which is shown by this dotted line in the diagram. So it needs to dissolve so the DNA can move about in the cell, staying within the cell membrane, to create those two daughter cells. The next stage is known as metaphase. And during metaphase, chromosomes are moved to the middle of the cell. It's an easy way to remember what happens in metaphase because middle and meta both begin with an M. They're pulled by structures called spindle fibers, which are attach themselves to the centromeres in the middle of the chromosomes. The sister chromatids are still attached, and then they are attached to the spindle fibers, as the diagram is trying to show you, with these strands representing the spindle fibers, and there would be one going to the chromatid on one side, and from the other side of the cell, this spindle fiber will be going to the chromatid on the other side. This prepares for the next stage, which is known as anaphase. Anaphase, remember, means apart. That was one of our vocabulary words. During anaphase, the centromeres split apart and the chromatids are separated. So one copy of the DNA heads to one side of the cell and one copy heads to the other. They're pulled apart in opposite directions. Because the centromere is sort of in the center of the cell, the chromosomes kind of take on this V shape because they're being pulled here from the center. And then you have telophase. During telophase, you have the DNA that's been pulled apart and is collecting at either end of the parent cell. The nuclear membrane will reform around those chromosomes, and the chromosomes will decondense. They will uncoil, forming chromatin again. Then the last step is for the cell to actually do the division into two identical daughter cells. In animal cells, the cell membrane pinches in. So this diagram is showing this membrane starting to pinch in. We call that cytokinesis. In plant cells, because they are encased in a hard cell wall, that little bit of new cell wall has to be built in the middle. It's known as a cell plate because it really can't pinch in. The cellulose, it's too stiff. So here are some actual stained slides of some onion cells that are undergoing mitosis. This whole first section, these first two slides are showing you cells that are in interphase. You can see the chromatin basically is staining all one color. You can't see any distinct chromosomes. There's a very clear nuclear membrane. The nucleus is nice and round, tightly packed. So the cell is busy just being a plant cell at this point. Then we move into prophase. So during this time period, the chromosomes will condense. So you can see how we're seeing some little black blobs, some distinct differences in coloration in the nucleus, and that the edge of this nucleus, is looking a little ragged because that nuclear membrane is falling apart so that the DNA will be free to move around the cell. After prophase comes metaphase, and in metaphase, your chromosomes are all lining up in a straight line, roughly in the middle of the cell. So here's sort of the outside of this cell, and down along the middle, we have all the chromosomes lined up. We can't really see the spindle fibers that are pushing them around. They'd be going this way, but it's this sort of straight line across the middle of the cell that's a characteristic appearance of metaphase. Then in anaphase, the chromosomes are being pulled apart. So you can see how we've got this V shape here because the spindle fibers are attached to the centromeres and they're pulling them this way on this side, and this side is being pulled that way. And then finally in telophase or telophase, what becomes most obvious there is that you have smaller cells side by side. So two smaller cells are letting you know that this cell plate has been built in the middle. These two nucleus to either side are from the daughter cells that have been produced. I just want to talk briefly about these 2N or N, the chromosome numbers. The letter N is representing the number of chromosomes in one of those sex cells or gametes in sexual reproducing organisms. So the gamete itself is a N cell. It's a cell with N number of chromosomes or a haploid cell. In this illustration showing you have one copy of all, in this case, 3 different chromosomes for this organism. But all the other cells in the body that are not gametes or not sex cells, they will have a matching pair of chromosomes. So they are 2N because they're going to receive one from each parent. So these are our diploid cells. We see an illustration here showing you have 2 copies of each type of chromosome because one will come from each parent in sexual reproduction. So the N refers to really how many chromosomes are there when you're thinking about, is it a gamete, is it a sex cell, or is it one of the other cells that has a full complement of DNA? Every organism has its own specific number of chromosomes. Humans have 46 chromosomes, which are then arranged as 23 pairs. So you get 23 chromosomes from your mother, 23 chromosomes from your father, but all of your cells, except from your gametes, will contain 46 chromosomes. But other organisms have vastly different numbers. Your dog has 78 chromosomes, goldfish 94, lettuce 18. Ferns have over 1,000. Which is kind of amazing. And then, fruit flies only have 8. Along with different number of chromosomes, the amount of DNA, how much DNA there is just in how much mass of DNA, and how many genes are on that DNA also varies from organism to organism. And there doesn't seem to be a clear pattern, so scientists are not quite sure, why some seem so vastly different than others. But many of the genes found in the different organisms are very, very similar because we're all living on Earth, and there are very similar chemical processes that have to happen in order for things to stay alive. So you will have genes in your body that are similar to genes that are in dogs or that are found in lettuce or found in a fruit fly, just because we all are Earth creatures. I just want to end this video by reminding you that mitosis allows the transfer of DNA from a parent cell to a daughter cell. And DNA is important because it is the directions for making protein. And protein is what organisms are made of, and proteins are the kind of molecules that produce enzymes, and enzymes allow the chemical reactions that preserve life. RNA is an intermediate step that allows the information from DNA, where it's held safe in the nucleus, to move out into the cytoplasm, where it can be then taken up by ribosomes to create those proteins. But the whole thing about mitosis is preserving that DNA so that proteins can be made to keep organisms surviving.